Hi everybody, welcome back to Synthetic Biology 1. Today we're going to talk about proteins and how to express them. Proteins are the powerhouse of the cell. They are responsible for most of what goes on inside a living system. They can perform chemical reactions, replicate DNA, sense molecules in the environment, and even control the function of other proteins. So if we understand protein expression, we'll unlock many great new tools for genetic design. In synthetic biology, we try to think about protein expression from two different points of view. To a biologist, protein expression is like a cuckoo clock, with all of this precisely evolved machinery working together. It's taken generations of biologists to collect all of the details, and of course there's many things that we still don't know. We are biologists, and we love and appreciate biological complexity. But on the other hand, we're engineers, and we hate biological complexity, and we find it very annoying. I don't want to build a cuckoo clock every time I have to express a protein. I just want a small number of simple pieces that I can put together and get the result that I want most of the time. Now, the cost of this simplification is that sometimes we'll miss things. But the benefit is that we can free up brain power to think about other details of the design. Okay? Enough philosophy. Let's talk about parts. If you want to express a protein in bacteria, you need to assemble a DNA sequence with 4 parts. Number 1, the coding sequence. This is the heart of the protein coding gene. It's the DNA sequence that codes for the amino acid sequence of the protein that we're expressing. The amino acid sequence of the protein, in turn, determines the structure of the protein and therefore the function. A coding sequence always has a start codon, ATG, and a stop codon, like TGA. In between, you expect to see enough codons to make a reasonable protein, like 100. You can always recognize a coding sequence by these features, or even better, your DNA sequence editor software can do it for you. Number 2, the promoter. If the coding sequence tells the cell what to make, the promoter is there to tell the cell how much to make and when to make it. The promoter is the place where RNA polymerase binds and begins the process of transcription, reading the DNA sequence and producing a matching messenger RNA or mRNA. Number 3, the terminator. Just like the promoter tells the RNA polymerase where to start, the terminator tells it where to stop. It's a special code that tells the RNA polymerase to stop transcription and fall off the DNA. Sometimes you'll see people leave the terminator out because the RNA polymerase will eventually fall off the DNA anyway, but including a specific stop signal, you don't have to guess where your construct ends. Number 4, the ribosome binding site, or RBS. Now that the mRNA is produced, it needs to be translated into a protein. The ribosome is responsible for this process, and the RBS tells the ribosome where to bind and get started. The ribosome has a particular role to play in determining how much of a protein is expressed. We say an RBS is strong if it binds tightly to the ribosome, allowing many ribosomes to produce many copies of the protein. The ribosome also has a specific stop signal. In this case, it's the stop codon of the CDS that tells the ribosome when to fall off. We don't have to add a specific translational terminator because it's already included in the CDS. And that's it. 4 pieces of DNA, always in the same order. Promoter, RBS, CDS, terminator. Each piece works separately from the others, and each has a well-defined beginning and an end. Those are the properties that we're looking for when we call a DNA sequence modular. It makes our lives as DNA designers so much easier. You don't have to reinvent a promoter for each new protein that you want to express. In fact, you'll find yourself using the same promoters over and over again until they start to feel like old friends. It also makes our lives easier as students of synthetic biology. we can separate out and focus on each individual element to better understand how it works and how we can control it. This view of gene expression doesn't include every detail of the biological cuckoo clock, but most of the time it gets the job done. Okay, until next time, everybody. Cuckoo!